Our first speaker in this post-coffee break session is Paul Ensel, who's I'm not sure the official title, he's kind of deputy chair, vice chair, or organizer of committees, or various things like that in pharmacology. And he's going to be talking to us about GPCR expression in health and disease. So, Paul.
Thank you, Andy. But there's a subtitle which is Using Lessons from Palmer-Taylor. And I've actually come up with, I think, some words that fit with the letters of Palmer's name. And those of you who know Palmer will, I think, hopefully agree with me about most of these. So P is for polite but proper level of skepticism is a necessity. Always consider the kinetics of response and reactions, kinetics of reactions. Let the literature guide, but not stifle you. Make sure to stay abreast of new techniques and discoveries. Education of students and colleagues is a high priority. And remember to think outside the box. And so those are the lessons from Palmer in science. And I'll have some other lessons as my talk proceeds. And you know, yesterday's session was about toxins and cholinergic, nicotinic cholinergic stuff and acetylcholinesterase, which has been Palmer's primary love. But some of you may not know, but a few of us remember, that Palmer actually worked on GPCRs, albeit it was mostly 25 years ago. And I found a whole series of papers in searching that extend back from early work, which I actually, he and I actually were involved in together, but a number of other people in the audience, including Gabby and Joan and even Roger Chen, were co-authors with him on various studies that related to receptor signaling and receptor activity as related to adrenergic and other receptors. And so, you know, I would say he's sort of a closet GPCR guy. He's not really an official GPCR guy. And it's sort of strange that he has this long gap in activity from the late 80s till now, but maybe it's coming back. So what I'm going to talk about today is really three topics, rather briefly. And the data I'm going to show is all unpublished new data in a new area that I thought I could share with old friends and new friends. I'm going to give a quick overview on GPCRs, especially their role as drug targets. I'm going to then talk about how does one identify the GPCRs to target. And then I'm going to focus on a topic which hasn't been discussed, certainly not much in this session, a little bit by Joan, but very little in the larger world as well, which is GPCRs in cancer, which I think is a therapeutic opportunity that's been largely ignored. So as in this sort of busy slide, we realize that G-protein coupled receptors are this largest superfamily of membrane signaling receptors that recognize a large number of different kinds of chemical entities. And they are both chemosensory, meaning they respond to smells and tastes and light, as well they detect them. But there are also receptors from what have been called non-chemosensory receptors that recognize a large number of endogenous ligands of many different chemical entities. And it has this canonical structure, which I think the next speaker, who's going to be Roger Sunohara, is going to talk about in great detail. And they link to these heterotrimeric G proteins of the various classes that Joan Heller-Brown indicated in her talk, as well as to Bayer-Restin, another scaffolding protein. And both of those contribute to signaling by G-protein coupled receptors. Now, when I say that they're the largest family, there are actually 800 human receptors, and there are about 360 of these non-chemosensory receptors. But what's really important, and where I'm going to really dwell on toward later in my talk, is there are about a third of these receptors are orphan receptors, meaning we don't know who are their physiologic ligands. And so even though there's been a lot of work and a lot of drugs, as we'll see on GPCRs, there's still lots we actually don't know, and which is what keeps some of us still going after many years, as well as we hope will inspire students and other trainees. Now, there are these GPCR families that are based on sequence similarities, and I don't think you can see this very well, but of the class A receptors are known as the rhodopsin-like receptors, and they're the majority. But there are also these other families, class B, glutamate, class C, the secretin family, adhesion receptors, a topic of much current interest, and the frizzled family of receptor as well. So why are GPCRs such useful drugs? Well, first of all, when I say the evidence for that, it's from this particular study that was in Nature Drug Discovery some years ago, but other more recent studies show the same thing. About a quarter of all approved drugs work on GPCRs. And you say, well, why do we choose GPCRs? Why are they such good drug targets? Well, as I say, it's because of a number of factors. Number one, they have tissue selective expression. They're not expressed everywhere all the time, but they're in particular tissues. They're able to amplify those signaling pathways that I showed on an earlier slide. But I think one of the most important things is its accessibility from the extracellular space. There's surface receptors, doesn't have to cross the grease of the plasma membrane and go inside the cell usually, although we saw some evidence earlier about an intracellular GPCR, and so they may signal intracellularly. They interact with this very large family of chemicals, lipids, peptides, amines, fatty acids, and so forth. They're highly druggable, and importantly, they're generally safe as drug targets. So there's a lot of reasons why one might like to invent or discover additional GPCR drug targets. And in fact, they're still very popular. In 2014, 9 of the 41 FDA-approved drugs had GPCRs as their primary mechanism of action. But in fact, only about a small fraction, about 15%, have as yet been successfully targeted. So there's lots of room that suggests the potential for many additional GPCRs, particularly I'd like to point out orphan GPCRs. Orphans are defined as not knowing physiologic agonists, but maybe we can still make drugs to them even if we don't know what activates them physiologically. So how does one identify the GPCRs as targets? You've got lots of options. How do we decide what should be targeted? And the question that we're going to focus on this morning, that I'm going to focus on, is there evidence, for example, by using unbiased methods that can identify and quantify GPCRs in individual cell types, that GPCRs that we are currently targeting are really the optimal ones to target? And in fact, are there individual cell types that express GPCRs that have been missed as targets and might be important physiological regulators as well? And further, are there disease cells, changes in disease cells in which expression of these receptors might be increased, and which would favor their potential use as drug targets, or if they're uniquely expressed? I'm going to show you some examples where I think that may be the case. So, in fact, it could be said, somewhat cynically, that what we've been doing, we in the larger sense, not me personally, but not the royal we, but we as a community who study and work on GPCRs and develop drugs and treat patients with GPCRs, drugs directed at them, we're looking for the GPCRs, the keys under the streetlight. And you all know, I think, many of you know the old joke about the drunk who lost his keys and was searching for them under the streetlight. And somebody comes along and says, well, where did you drop them? And he says, well, over here. So why are you looking under the light? Because the light is so much better there. And so the question is, perhaps those of us who have worked on these receptors and who develop and use these drugs, perhaps are we akin to this drunk? It's an open-ended question, rhetorical question, if you will. So my lab has actually devoted substantial recent efforts, mostly unpublished so far, to answer this question by assessing GPCR expression in cell types that include a wide variety, including tissue fibroblasts, iPS cell-derived cardiac myocytes, adipocytes. Papers describing these are in press in a couple of journals. Neurons, one paper came out a few months ago with people from New Zealand. Vascular smooth muscle cells, lots of blood cells, but also in cancer cells. And the cells that I'm going to focus on today are pancreatic ductal adenocarcinoma cells. I'll tell you about why in a minute. And cells in the tumor microenvironment. In particular, the microenvironment that nurtures and collaborates with primary cancer cells. And in particular, what we focused on are cells known as cancer-associated fibroblasts, or CAFs. So how would you study this? Well, somewhat dated, I guess, is the use of these generic DNA arrays, such as ones made by Affymetrix and others, these commercial arrays. But it turns out, and I'm not going to have time to show you the data, these are not optimized to detect GPCRs for a variety of reasons, which I'm happy to discuss later. There are some proprietary GPCR microarrays and other methods, such as multiplex PCR, but that hasn't been widely used, of course. And large-scale whole genome analysis, such as RNA-seq, which we and others are starting to use for that. But of course, you generate a large amount of data for other components that you don't necessarily care about. And so what we have focused on has been the use of real-time PCR, GPCR arrays that can assess and quantify virtually all, or about 90+% of the non-chemosensory GPCRs in rats, mice, and human cells. Very simple assay. It works by making a cell isolation, preparing RNA, cDNA synthesis, hybridizing to the array, generating data, and then trying to figure out how to classify the various GPCRs that you find, which we've done by primarily by classifying them and whether they link to GS or GI or the GQ11 or G1213 or as orphan receptors, which of course can also have such linkages. So I'm going to just give you some conclusions from the general work that we've done without going through any data on this part, because I'm going to focus on the cancer data for the remainder of my talk. Our results have actually revealed that many cell types, we've looked at about 15 different cell types already, all express about 100 GPCRs on any cell type. Now, we haven't looked yet, we're just started setting up to do individual cells, but on individual cell types, we're talking about more than 100 GPCRs. And we've done independent qPCR for lots of the data that we've generated. It almost always confirms what we see on these arrays. We've done preliminary RNA-seq data. It confirms what I'm going to show you. But the most important conclusion, which I've bolded here, is that the most highly expressed receptors are generally not ones that were previously studied, or for which there was very much, in some cases, no prior data have existed. So we're this is discovery talk. I'm telling you about discovering something that nature has decided we should choose. She chose this, we didn't choose this, but most of us have ignored listening to nature because we studied what we wanted to study. So what we think is, and what we've shown is that a number of these newly recognized receptors are functional, they can regulate cell physiology, and they can show altered response in disease cells and may contribute to pathophysiology. So our bias, what we're working on actively, people in my group, are what we're calling GPCR omics. We have to have an omics term. Unbiased GPCR profiling that can identify these receptors not previously known to be expressed, that can be functional, and comparing healthy and diseased cells, we think can identify novel therapeutic targets, including orphan GPCRs. So I'm going to turn to one subclass of the data related to cancer. So should we consider GPCRs in cancer? Now, in a few cases, GPCRs are mutated in cancer and might be driver mutations. Work by Silvio Gutkin and others have indicated that. And it's clear that the growth and function of a large number of endocrine gland tumors, which are driven by hormones that work on GPCRs, pituitary, adrenal, thyroid, ovary, testes, a number of those are clearly, one could think about GPCRs as drivers of aspects of the malignant phenotype. But, and some, as I've said, some data from the Cancer Genome Atlas and elsewhere suggests that there are mutations. But frankly, there have been many recent reports that have really argued that GPCRs are largely off the map in cancer because they haven't been perceived in a primary way as the leading driver of mutations. There's lots of reasons for this, and a couple of recent reviews have commented about this, but as one stated, GPCRs represent a highly druggable yet understudied molecules in cancer. And so we really asked, are there GPCRs in other cancers that might be therapeutic targets, perhaps allowing biomarker-guided precision medicine approaches for their treatment? And so We also think that it's worth considering it may not be GPCRs in the cancers themselves, but tumors are comprised of multiple cell types in addition to the cancer cells. And so we're asked whether GPCR expression on cells other than the cancer cells, for example, on cells of this tumor microenvironment, might be important. And we're particularly interested in cancer-associated fibroblasts, also called stromal cells, which are a very important cell type in the tumor microenvironment. They're particularly important, these CAFs, in tumors that are known to be highly fibrotic. The word that's typically used for that is desmoplastic tumors, such as pancreatic cancer in particular. Well, what about pancreatic cancer? Probably many of you don't know much about it, although if you unfortunately suffered the fate of I have of knowing family and friends who've suffered with this disease, you become acutely aware of its incredible lethality. It is the fourth leading cause of cancer-related deaths in the United States at the present time. Because our progress in its treatment is so bad, it's predicted to be the second leading cause of death within the next 4 years. And there are about over 300,000 cases of this disease around the world. There have been no major improvements in its therapy over the last 40 years. About 6% of people who have this cancer live beyond live 5 years. So over essentially 95% of people. This is a diagnosis of you're going to die reasonably soon. And many of us know our good friend and mentor Al Gilman passed away with this disease about 3 weeks ago. And so, it's touched many of us, I think, quite closely. Most patients are diagnosed at an advanced stage, with only about 15% eligible to get surgery, which is the one treatment that's considered curative. And resistance to treatment is common, and in part it's linked to this desmoplasia, this fibrotic stroma within the tumor. So, what is it about this? How does pancreatic cancer work? So this is a pancreatic duct. These are the acinar cells that produce enzymes and fluids that go out into the duct and that are a part of our digestive network. These are and the cells in between them in brown are known as stellate cells, these supportive stromal cells. And during the progression from essentially an in situ sort of early stage tumor, intraductal tumor, one sees the progression to these invasive cancer cells, and that happens in these ductal epithelium. But in addition, you see this proliferation and activity of the stromal cells to create a large amount of extracellular matrix. So what actually happens is these cells can promote tumor development as well as maintain proliferation, survival, migration, invasion, and they can promote immune suppression. And they're involved in the resistance to chemotherapy. And so these are the picture here of a CAF that essentially can influence the immune cell, the tumor cells, makes extracellular matrix, and there's this collaborative interaction between these cell types. So we've asked the question, as you can tell from where I've taken this so far, might GPCRs in the cancer cells and or in the pancreatic CAFs contribute to the malignant phenotype? And could they be therapeutic targets? And what I'm going to show you is preliminary data that says the answer to those questions is yes. And I'll show you just a bit of the data that we have. And it's still pretty early days, but I think it's potentially exciting and important. And it's a direction that we're taking very actively because I think we have a chance to really make a difference in this very lethal disease. So what we've done was first to take out some pancreatic tumors in collaboration with Andy Lowy, work that was done by a master's student, Talia McCann. We took out he had 3 tumors, and we did a GPCR array on the whole tumor. And what we found were that these tumors had a large number of GPCRs, as I've told you, well over 100. But if you looked at the GPCRs that were shared among them in these tumors, it was about 78 that were readily detectable. However, if you looked if we compared not the tumors, but if we actually took primary cultures that were created by Dr. Lowy's lab, along with established cell lines. So we took patient-derived samples here, given numbers 34E and 7090, as well as established cell lines that can be obtained commercially. and we did all this GPCR array analysis, those are what all the numbers are, there were essentially 46 GPCRs that were shared in all the samples. That doesn't mean some of the others aren't important, but these are the ones, the common receptors that were found in all the cells. And in particular, what I want to focus on is that we found 2 receptors that were prominently upregulated in, that we've called GPCR A and B, that were prominently upregulated in the cancer cells, and we compared their expression to normal pancreatic ductal epithelium, or what's called PDAC cells. And these are values for their elevated increase in expression. And you can see this one orphan that I'm going to tell you about has a delta CT, which translates to about a 30-fold higher expression in the cancer cells. And so between 16 and 30-fold increase. And as I already mentioned quickly before, this is qPCR data compared to array data on the left on the array, right is qPCR. And this is just looking at these delta CTs, where a higher delta CT means lower expression. Here's the normal pancreatic ductal cell. Here's these 2 patients, and here's the cell lines. And in all cases, there's a higher expression that we can detect. Moreover, if one goes to the literature and looks at a group of patient samples, that these are not our data. We actually went to a geoset in the literature, and if you just look at the red bars, this is the expression of this particular orphan receptor in a large number of pancreatic tumors. This is the expression in normal pancreas. So we believe that this receptor seems to be a receptor that is at least much more highly expressed in pancreatic cancer than in normal ductal epithelium. Moreover, if we transfect this receptor into a normal pancreatic ductal epithelium, we increase DNA replication roughly 2-fold. So it seems like it may be functional. So those are some data on the cells themselves. But what about these cells? As I've mentioned, we've got data here now that there's possibility of an orphan here, but what about on these cells in the tumor microenvironment? And I've already told you a little bit about these calves, but just another picture to make this even more prominent. So here's the pancreatic ductal epithelial cell. The precursor cells for these calves are known as pancreatic fibroblasts or pancreatic stellate cells. They have a number of features that I'm not going to review, but there is this collaborative relationship between these 2 cells. And these cells are really thought to be activated fibroblasts. They express a large number of fibrotic markers, including alpha smooth muscle actin and several collagens and others. So what we've asked is, might these calves have preferentially expressed or increased the expression of GPCRs, and might they be functional, and might they be therapeutic targets? Well, just to confirm what I've just told you is the case, this is the expression of alpha smooth muscle actin in pancreatic fibroblasts, normal pancreatic stellate cells, and in primary cultures that were established from 5 different patients with pancreatic cancer. And you can see a prominent increase in the expression of alpha smooth muscle actin, suggesting that these are profibrotic cells. When we then did these array analysis of these 5 samples, we found that they actually shared 102 GPCRs in their expression, and they fell into these various categories in terms of whether or not they linked to GI, GQ, G11, or others. But about 40 of them were actually unknown. Importantly, though, of these 102 receptors, 36 of them were orphan GPCRs. So about 35% of the receptors were actually orphans. Moreover, when we compared the expression in these 5 patients versus normal pancreatic fibroblasts or pancreatic stellate cells, these precursor cells, we saw that there were about 35 receptors, 10 of which were orphans in this group, and 12 of which were orphans in this group of 33. Very rarely were the receptors down. They're consistently increased. And one of the receptors, which has had very prominent and consistently high increased expression, is one that we've called GPCR orphan 1. And in fact, independent mRNA analysis of these calves compared to the PSCs and PFs showed very prominent increase in expression of this orphan at both the RNA and at the protein level in all 5 of these samples. And when we actually got an antibody to this receptor, we stained tumor samples from patients, and this was done by a former postdoc who's a pathologist in Japan. You can see this very prominent expression both in the interstitium, but actually also on the tumor cells themselves, which we have other data that support that. Importantly, if we transfect this receptor into normal pancreatic stellate cells, a series of fibrotic markers, alpha smooth muscle actin, a collagen, interleukin 6, periostin, and CTGF, CCM2, all go up several fold. And if we use siRNA to knock down the expression of the orphan in the calves themselves, we decrease the expression of smooth muscle actin, IL-6, and IL-6. So what I've tried to show you is that GPCR omics by using these GPCR arrays, is useful for identifying and quantifying cellular GPCR expression, that GPCR omics of healthy and diseased cells can reveal receptors that can contribute, we think, to pathophysiology and be therapeutic targets, including for diseases such as pancreatic cancer, which are in desperate need of new therapies, and that certain GPCRs, including orphans, whose expression is increased in these cells, such as the ones I've showed you, orphan A in the PDAC cells, orphan 1 in the calves, can regulate features of the malignant phenotype. So the results overall imply that GPCR omics of healthy and diseased human cells can identify GPCR targets in cancer cells, of course, as well as potentially other diseases, and cells of the tumor microenvironment, and very, very likely may be contributing to a function and have the potential to be useful targets. I'd like to thank the people who did the work that I've told you about. I forgot to mention Sue Chow, who's the postdoc who did most of this work, along with Atalia McCann, together with Andy Lowy, and his postdoc Randy French, and Sarah Chang, another graduate student, and others in my lab who are involved in these funding sources. And lastly, I want to complete Palmer's name. I've told you about Palmer in science, but what about Palmer in life? What are the life lessons from Palmer? Well, take every opportunity to learn something new. Avoid anger at all times if possible. Those of you who know Palmer know what I'm talking about. Young people are the future. Nurture them. Lubricate with fine wine to promote collegiality. Once you commit to a task, make an impact. And last but not least, remain a loyal friend and mentor. Thanks for your attention. And thank you, Palmer.
Questions?
So, great talk, Paul. Really exciting and intriguing story. I'm curious about these receptors you've identified in pancreatic cancer and what's known about their expression in the rest of the body if they pop up elsewhere.
Sure. That's that. I didn't have time to go through that. So the orphan receptor that's present in the cancer cells has a relatively restricted expression. The other receptor has also had a somewhat restricted expression, but it's been found in a number of disease settings. We've actually pretty much figured out what we think the ligand is, and I'm not at liberty to discuss that yet, but it fits very nicely with why it would be present in cancer in terms of the ligand that's there. Palmer.
What do you think is more important when you identify one of these entities, one of the GPCR, to go upstream and identify the ligands, or to go downstream and identify the pathway?
Yeah, so that's a great question. We've wrestled with that. The way I've been focusing this in my thinking, Palmer, is you have to sort of do both, but I'm really trying to push this project toward a therapeutic approach. So we're going to be hopefully screening for some drugs for these receptors. And even if we don't know exactly how it signals, or even if we don't know exactly the ligand, that doesn't mean we can't come up with an antagonist that would block its function. In addition, we are trying to develop biologics such as antibodies and nanobodies that might be created to these receptors that could potentially block their action. And so although as a scientist who wants to understand mechanisms, it would be very important to answer all the downstream stuff. I think if we could block these and we can show they're really playing a role, they're not going to be the whole story, no one of them. Nor, I think, is it going to be the case that you would avoid perhaps even traditional chemotherapy. They may be an adjunct to that. But if GPCRs are playing a role on multiple cell types in there, or there are multiple cell types involved that we can target, with respect to metabolism, with respect to migration, and some of the other features that go on in cancer, I think we can potentially make an impact. And, I've purposely picked a disease that was really tough because I think this is something a challenge worth taking on.
One more question from Jean-Pierre.
Jean-Pierre.
Are you in a position to establish causality and correlation? In the sense that of some of the effects.
Yeah, so no, that's a great question, Jean-Pierre. I mean, the question is, are these part of the malignant process that initiated it? Probably not. But are they ones that might continue to keep the progression going on? Probably so. So that's how I see it. They're not the thing that started it, but they may be contributing to the ongoing features. What?
None of them.
I don't know. We don't have it. Well, there's data that in some cases there are a few GPCR mutations that have been implicated, but it's not. It's too early to say. It's too early to say. Really don't know. Susan?
So obviously it's a huge opportunity with Silvio being here. His work on the GS proteins, one step down, EKA the next step down. I think it's a huge opportunity, and I think the stellate cells, the surrounding cells, and what are these tumor cells shedding? What's coming off in exosomes that is really driving? Perhaps the primary mutation is in the tumor cell, but then they're driving the surrounding cells and looking at tissues. I mean, it's something I really come to appreciate how important it is to look at the actual tissues, because you can't just look on a neuron in isolation. As the astrocytes and the oligodendrocytes, they work together, they're a team.
That's why I showed data in both sets for tissues, because we do have data on tissues. But let me just say about exosomes. I actually wrote a small grant this week on exactly that topic, somehow even during this meeting. But it turned out that I'm not a member yet of the American Association of Cancer Research, so they won't let me set it in. I'm waiting to hear if they'll approve my membership, which I had got sponsored the day before yesterday, and then I can put the grant in by Monday. So to do the exosome experiment.
Let's thank Paul for a nice talk. So the next speaker who is listed on the program is Art Christopoulos, and unfortunately he had some back trouble that he's battling that prevented him from taking a long flight from Australia here. But we're very fortunate to have Roger Sunahara here, who is extremely exciting in talking about GPCRs. I don't have an exact title, but I'm sure it's GPCRs and probably the G protein and other interesting things. So Roger.
Thank you. Well, first I'd like to thank the organizers for allowing me to fill in for Arthur. Arthur, as Andy said, has a chronic back problem. For as long as I've known him, he's had this back problem, and it was just insurmountable in terms of his pain, and he apologizes profusely for not being able to make the talk. I will talk about allostery, as Arthur would have. Unfortunately, I don't have the same cool Australian accent that he has, and I don't have a million really, really cool data slides on allostery that he does. But what I will tell you about is some stuff that we've been working on for a long time, and it's been very close to my heart, and that is trying to figure out how G proteins get activated by G protein coupled receptors, and in turn, how that activation process affects the behavior and the conformation of the receptor itself. And so clearly that is allostery. And this symposium has been amazing to see the trainees, as many of the speakers have commented that Palmer and Susan have had. And I have to say that, so I was at University of Michigan for 14 years. I just moved my lab here to the University of California, San Diego in July. Michigan tried to recruit Palmer and Susan many years ago. And I figured, okay, well, they didn't come, so I might as well come to where Palmer and Susan are. But what is highlighting, obviously, their careers, and Palmer's careers, really scholarship, mentorship, leadership, and being awesome scientific and community citizens. And Palmer reminds me very much of a very close and dear friend of mine that Paul just mentioned. He was my former postdoctoral advisor, Al Gilman. If many of you don't know, on December 23rd, he succumbed to cancer. And many of you obviously know him because he was the editor of, I think, 5 editions of the Goodman and Gilman pharmacological basis of therapeutics, and also won the Nobel Prize for discovering, along with Marty Rodbell, G proteins. And Al has certainly been an inspiration in my career and been a great mentor, a great scholar, leader, and citizen like Palmer. So it's really interesting to be in this field. It's a rather older field. And I like this slide. This is a rendition taken from Earl Sutherland and Ted Rawls' version of how hormones can regulate protein kinase A, and in fact they were interested in glucagon-stimulated glycogen metabolism. And back in 1957, it was thought that the cell surface receptor would recognize a hormone, either glucagon or epinephrine, and make cyclic AMP. This is in 1957. So since I've been in science and been in research, I've been fascinated by this simple question. How does this actually work? What does it look like? How does it work? And so, fortunately, I get to follow Paul, and he was, when he was giving his talk, I got to take all my slides out one at a time. Thank you, Paul. Thank you, Paul. Thank you, Paul. Just to say that there are many GPCRs, as you know, hundreds of them, and they're fantastic therapeutic targets. But the thing that's cool about GPCRs is that despite their difference in primary sequences, despite their capacity to recognize and be activated by different stimuli, they all seem to have the same capacity to activate, or most of them at least, activate G proteins. So whatever is happening on the extracellular or outside of the cell, however different they are, they can all converge on a common mechanism on the intracellular part of the cell and activate G proteins. And so, out of all the GPCRs, hundreds that we could have chosen, I decided that my lab, we would focus on the beta-2 adrenergic receptor. Why? It was one of the first GPCRs to be cloned. It was one of the first ones to be purified. It's a fantastic therapeutic target so there are lots of drugs and reagents, and it's been heavily studied through mutagenesis. So I figured, well, if we're going to study it, we can gain from other people's knowledge and their mistakes, and try to figure out how this system works. So we focus on the beta-2 adrenergic receptor, which couples to the stimulatory G protein GS, which obviously stimulates adenyl cyclase to make cyclic AMP. And although the beta receptor, like many GPCRs, can couple to other G proteins as well as interact with other interactors, which have the capacity to regulate signal transduction pathways, including transcription, I'm going to really focus my talk on how the beta-2 adrenergic receptor talks to its cognate G protein GS, the stimulatory G protein GS. And I will use it as sort of a model to how we think that the beta receptor, or all GPCRs, couple to their cognate G proteins. And so before I go into that, I'm going to go back to a sort of a Goodman and Gilman rendition of the G protein cycle, and how hormone activation of these receptors activates this whole pathway. So it starts at this very early step where hormone binding to the cell surface receptor acts and interacts with the receptor, tickles a G protein, and does a very key step, And that is to tell the G protein to knock off GDP. That's all the receptor does in terms of G protein. Because as soon as GDP falls off, the high concentrations of GTP immediately bind. And that binding event will stabilize a conformational change in the subunits in what we like to term as a functional dissociation of the subunits. And functional so that they can interact with effectors. And in this case, I've just highlighted adenylyl cyclase as one of the effectors for the alpha subunit, the stimulatory G protein, GS. Now, G proteins are not only guanine nucleotide binding proteins, but they're GTPases. They can convert GTP to GDP. And that conversion turns out to be a nice little timing mechanism, because as long as it's in the GTP bound form, it binds to cyclase very well, it binds to effectors very well, and makes cyclase turn over lots of cyclic AMP, or ATP. But as soon as it's hydrolyzed to GDP, the affinity for the alpha subunit is decreased, and in fact would prefer to rebind beta gamma. And then, of course, you end up at the top of the cycle again. But as you notice, one of the key regulators in this whole pathway is this step up here. How does a hormone, when it binds to the receptor, stimulate the G protein and tell it to lose nucleotide? And this process is basically what we've been studying in the lab for the last 20 years. And we've taken advantage of a lot of developments in the field, but primarily the use of biochemistry, biophysics, and in particular X-ray crystallography. And we've tried to use structure to help us understand all these processes, the conformational changes, the protein interactions, and even to develop ligands that can modulate these pathways. But for many, many, many years in the field, what was missing was any structural information that could help us understand this very important step in the process, and that is this receptor G protein complex. Until recently, a long-standing collaboration with Brian Kubilka's lab at Stanford, we were able to, in fact, solve this structure. And it came out a number of years ago. So, allostery. So we know that hormones can activate G proteins, and we, as I just said, what their primary function is to knock GDP off of alpha subunits, and we can measure this activation clearly by measuring radioactive GTP gamma thiol binding, and you can see it occurs over a agonist-dependent concentration curve or pathway. And as well, you can measure downstream activation of GS and see how it interacts with adenylyl cyclase, again, being stimulated in an agonist-dependent manner. So, yes, hormones can obviously activate G proteins. But once they're activated, G proteins can also affect the conformation of the receptor very tightly. And it turns out that this allosteric effect of the G protein on the receptor has been known for years, studied for years. And Jean-Pierre Changeux has used this allostery as one of the primary examples of really cool receptor allostery pharmacology. And it was, this is one example of this allostery that was measured using classic pharmacological assays. This paper was published in the 1980s by André Delion when he was a postdoc or working with Bob Lefkowitz, but really is representative of a series of studies that were done in the late 70s. And what André Delion did is he used an antagonist probe to bind to the beta-adrenergic receptor, and he tried to compete binding with an agonist, isoproterenol. And what you may notice is that the competition was not one site. It actually spanned several log units, 4 log units. But the cool part of this experiment is that when he repeated that, but added in non-hydrolyzable analogs of GTP, GPP, NHP, and repeated this competition curve, he completely changed the shape of the competition curve. What he did was he uncoupled whatever it was that produced this high affinity binding with a non-hydrolyzable analog. So, in the absence of guanine nucleotides, it seems that isoproterenol can inhibit in two states, a low state and a high state, But as soon as you add in guanine nucleotides, you shift it all to a low state. And so the term at the time, during the late 70s and 80s, was that this high affinity state agonist binding was in fact the G protein coupled state. That was the drug agonist receptor G protein complex, or the ternary complex. And then if you uncouple the G protein from the receptor, then agonist bound with very low affinity. And as really, I've been studying this figure for 25 years. This one figure, trying to figure out how does this thing work. So this is, as I said, the long-standing collaboration with Brian Kobylka's lab, and we've been trying to figure out how this thing works. And what I'm going to tell you about is a little bit is about the structure that we've been able to solve to help us understand this, as well as lots of pharmacology based on the models we've generated on our structure. Of course, we were able to solve the crystal structure of the beta-adrenergic receptor bound to the heterotrimeric G protein. This is the alpha subunit in yellow, beta gamma in cyan and sort of a purplish color. And most importantly, the G protein is nucleotide free. We had a lot of tricks. We had nanobodies, which stabilize this complex. We used fusions to T4 lysozyme to help in packing. But nonetheless, we were able to solve the crystal structure. I'm not going to go into a lot of details about the crystal structure. I'm just going to really highlight major features. And one of the most interesting features, but not surprising, was the heterotrimeric G protein alpha subunit interacted with the receptor through the C terminus of the G protein. And this was known because peptide studies and site-directed immunogenesis studies, primarily from Heidi Ham's lab, had really identified the C terminus of alpha subunits being the prime interacting part of the receptor. What came to us a little bit by surprise, was the interaction with the N terminus of the G protein and how it interacts with the receptor. And it turns out that interaction is very, very important for activation of G proteins. I'm sorry I don't have time to talk about that, but it is critical. What I'm going to focus really on is on the C terminus and what the C terminus does. Okay, so at the time that we were trying to solve the structure, Ron Dror, when he was working at D.E. Shaw in New York, D.E. Shaw Research Institute, did a really cool simulation. He did a whole atom, long time scale simulation of the beta-adrenergic receptor, trying to figure out how ligand binding occurs. And so here what he was doing is trying to simulate alprenolol binding to the beta-adrenergic receptor. Now at the time, we didn't have a structure of the active conformation of the receptor. We only had a structure of the alprenolol bound, or the inactive receptor. And I want you, if you can, pay close attention to how alprenolol is binding and the steps at which it's binding. Whoops. So you can see it sort of bounces around and sort of sits at the top of the receptor, hangs around, and then dips into the site. So we're going to take a closer look at this. And what I would like you to do is, if you can, focus on these two aromatic residues. There's a phenylalanine and a tyrosine. It's not really visible here in this view, but you'll see them. And you'll see that alprenolol, when it sort of hangs around this little vestibule, it sits on top of these two aromatic residues until they almost move out of the way and then dip into the actual binding site where the hormone binds. And so this wasn't the goal of Ron's study. It sort of came out afterward, that these two aromatic residues are almost forming a lid over the orthosteric binding site. And that lid must be important for something. And what I'm going to tell you about is a model that we've now created or generated that uses some of Ron's ideas and also uses all of what we know about the pharmacology of GPCRs to help us explain André Delian's observation and others in the 80s. So what we think is that receptors are normally exist in their what we call open inactive state. So they're open in that they can allow ligands to bind very rapidly, but also remember that they can dissociate very rapidly. And they will bind with low affinity. And really, we think that this state of the receptor is what André de Leon and others have observed as their low affinity or uncoupled state. But because in this case, the ligand is an agonist, it obviously has some property that changes the conformation of the receptor, which recruits a G protein and tells the G protein to lose nucleotide GDP. But what our crystal structure was helped us discern is that when you lose nucleotide, it changes the structure of the receptor and stabilizes what we call the closed conformation, closed active conformation. And obviously, by stabilizing this closed conformation, you now affect isoproterenol or agonist dissociation. You affect its off rate. And by affecting its off rate, all of a sudden you have something which binds with high affinity. And we think that this high affinity, or this closed active conformation, is in fact this high affinity binding site that André de Leon and others had observed. So GPCRs, we've been lucky to take advantage of crystal structures now of many GPCRs, going from the inactive to active conformation. And what we can sort of sum it up as this, is that when the C-terminus of the G protein interacts with the receptor, it stabilizes pretty dramatic conformational changes on the intracellular phase that are translated to the extracellular phase. And sort of a cheap comparison is sort of like a teeter-totter. So whatever happens on the inside really has an effect on the outside of the cell. And of course, if you think about our lid, it may stabilize this lid. Okay, so when we're after the crystal structure, and we are able to make these receptor G protein complexes, we decided, we have the coupled G protein coupled receptor. It's in this coupled state. How does it behave in a binding assay? And so a very talented graduate student, Giselle Valles-Ruiz, she took reconstituted purified beta receptor and threw in G protein and just incubated and cooked it in a test tube. And then looked at alprenolol binding. I remember when she did this experiment, she was at the binding, and I was ready with her, and she was so excited because this was going to be we were going to see 100% high state. Anyway, so the counts came off the counter, and this is purified protein. So she knew she put in approximately 30 femtomoles of receptor, 27 actually. Yet the first data to come off the counter said she had 18 femtomoles. And we thought, something wrong. She messed up the experiment. The second part of the experiment was, however, to measure alprenolol in the presence of GDP. So as soon as she put back in GDP, all of a sudden she saw the expected amount of binding. Then she decided, okay, well, if I take that GDP and get rid of it again, and in this case we used a non-selective nucleotide lyase to degrade all the GDP to GMP. And then when we did that and she repeated the binding assay, she could almost completely knock out binding of alprenolol. And then you could restore it again with adding excess GDP. And we were sitting, scratching our head, not being able to make any sense out of this. Why would nucleotide affect binding of an antagonist alprenolol? It makes no sense at all. We don't have any agonist. We have these huge effects on binding. And so Brian DeVries, another extremely talented graduate student in the lab, former graduate student, decided, okay, well, let's just take receptor and G protein and a priori get rid of nucleotide. A pyrase treat it. So this is a nucleotide-free complex. But in this case, do a saturation experiment with alprenolol. And you'll see that when he took this, he got, 6 femtomoles of binding. But when he uncoupled the G protein with GDP gamma thiol, he all of a sudden got 5 or 6 times the amount of binding sites. In fact, he got the number of binding sites that he expected for how much receptor he put in. And that you can not only do with GTP gamma thiol, but also you can do with GDP, and there's about a 10-fold affinity shift. So Brian was really intrigued by this. And of course, we were all scratching our head, how should this work, or why should this work? What is the basis of this appearance of binding sites? So Brian took the same preparation that was nucleotide-free, and instead of doing a saturation analysis, he simply did a time course. He looked at the on rate of ligands binding to this to the receptor, but he did it in the absence or presence of varying concentrations of nucleotide, in this case GDP. And what you can see is when you add in GDP, you really affect the on rate of ligands. Or you can uncouple it with GTP, gamma thiol, and get a maximal rate. So you dramatically affect the on rate. Now, as I said from the crystal structure, what it told us is that the C-terminus of the G protein is critical for its interaction with the receptor. And in fact, you can just make a fusion of the C-terminus of the G protein onto the C-terminus of the receptor and force the C-terminus up into the core of the receptor and arrive at the same entrée de liant high affinity agonist binding state. You don't even need the G protein. So what it says is that whatever occupies this site on the intracellular face of the receptor, it really affects agonist binding. So at the time we were doing this, we were very interested in other alternative ways of stabilizing active conformations of the receptor. And through a collaboration with Jan Steyer in a free university in Brussels, we were able to generate a nanobody, an antibody, camelid antibody, single chain, against the agonist-bound beta-adrenergic receptor and solve the crystal structure. This is actually Brian's lab, solve the crystal structure. But what was interesting about the structure, as it turns out, that the crystal structure of the nanobody-bound receptor, and this is in cyan and blue, is almost superimposable on the crystal structure of the G protein and the receptor in yellow and green. In other words, the nanobody is occupying the same space and almost doing exactly the same thing as the G protein does to the receptor, at least on the intracellular phase. So how does the nanobody behave in a binding assay? So what Brian did is he simply took the nanobody, took purified nanobody, and incubated varying concentrations and did the entrée de liant experiment. Of course, and he could completely shift the affinity of epinephrine, almost three orders of magnitude, shift the affinity. So really, the nanobody is essentially a G protein mimic. And Jake Mahoney, another talented graduate student in the lab, was able to use a different method to measure this receptor nanobody interaction, that is using interferometry. And what Jake was able to observe is a binding constant for NB80 to the naked receptor, unbound receptor. But if he did include isoproterenol, the agonist, he can shift the affinity for nanobody about 2.5 log units. So really, this nanobody is really like a G protein. And not only that, agonist binding seems to affect the on rate of the nanobody. So really, agonist binding is helping the receptor go into conformation that it can accept a nanobody or a G protein. So how can we use this nanobody? So Brian decided, okay, well, let's just take that nanobody, take what we know from Jake's experiment, and force the nanobody onto the receptor without a ligand. And then check what happens in our binding assays. So if our hypothesis is correct, alpranolol should not bind, or should bind slowly. So with no nanobody, you can see alpranolol binds very rapidly in a diffusion-limited manner, essentially. But if you pre-incubate the receptor with increasing concentrations and saturate the receptor with the nanobody, it really slows down the on rate of alpranolol. So this is really similar to the G protein effect I just described. So we thought this is cool and this makes sense. but is this an effect of an antagonist? Because we were using a neutral antagonist to hydroprenolol. How about other ligands? How about agonists? And so Jake Mahoney, and this took a long time to develop, basically demonstrated the same thing, that even an agonist binding to the receptor, its binding is inhibited by pre-incubation with nanobody 80. Moreover, he's saying that other agonists like methoxyphenoterol and even partial agonists can be inhibited by pre-incubating the receptor with nanobody NB80. Even an inverse agonist binding can be inhibited by NB80. So really, NB80 is stabilizing a conformation of the receptor that can't accept the binding of any ligand, regardless of its efficacy. Okay, so we've got this system going. We know the nanobody is really cool, and it seems to work just like a G protein. How about we try a slightly different experiment? We'll take the receptor, we'll pre-incubate it with alpranolol, and then we'll force on nanobody 80. And really, that would mimic the G protein in its nucleotide-free form. If our hypothesis is correct, what we should do is stabilize the closed conformation and dramatically affect the off rate of ligand. And that's exactly what happens. So here, with no nanobody, you can see alpranolol rapidly dissociates. But if you force on nanobody at very high concentrations, you can almost stop alpranolol from dissociating. And so by looking at the crystal structure, it really helps us understand how this works. So this morph is morphing between the inactive and active conformations of the beta-adrenergic receptor. Inactive bound to carazolol versus active bound to agonist and G protein. And you can see the hallmark activation is movement of TM6. This is the top view of the receptor. And look at these two aromatic residues, the same two aromatic residues that I was pointing at by Ron George simulations. And you can see when you go into the active conformation, these two aromatic residues really form almost like a cap or a lid on top of the orthosteric binding site. It's a lot easier to see in this surface representation. So, active, inactive, closed, open. And we think that this is really driven by the G protein. One, because we have all the biochemical data to suggest that it's driven by the G protein, but we're also able to take advantage of some crystallographic data, work from our lab compared to work from Chris Tate's lab at LMB in Cambridge on the beta-1 receptor. And what I've done is done the same simulation, is they were able to solve the structure of the beta-1 receptor bound to an agonist or bound to an inverse agonist. So what we can do in this morph is to distinguish what are the G protein effects, not just agonist binding, but what are the G protein effects. And again, I'd like you to focus on these two aromatic residues. Whoops. So really, the G protein conformation of the beta-2, which is in the yellow structure, is the structure which really forms this lid or cap over the orthosteric site. And now we have other receptor structures now. The mu-opioid receptor as well as the M2 muscarinic receptors. And we have inactive and active conformations. And so if our hypothesis is correct, we think that this conformational change going from the inactive to active conformation and formation of this lid will and should have the same pharmacological properties as the beta-2 adrenergic receptor. And if you do the assay, you can pre-incubate muscarinic receptor with G protein and then do a saturation with NMS, an antagonist. You can see that when you uncouple the G protein, you influence the rate of association of NMS. And of course, you can do the same experiment and make that difference or uncouple it with nucleotide GDP. What about other receptors that have much bigger ligands? The receptors I've been talking about have really small ligands and really tight binding sites. Mu-opioid receptor, however, binds essentially peptides. And its structure is very different, and it doesn't, as you can see in this morph between the inactive and active conformation, doesn't close over like the beta-2 or mu receptors. But what's interesting about it, what's interesting about the mu-opioid receptor is it also demonstrates G-protein-mediated high-affinity agonist binding, just like André Dillon had observed for the beta-2. And it is sensitive to nucleotide. So you can create high-affinity binding for an agonist like morphine. But how does that work if it doesn't really form a lid? So Jake decided to try the same experiment. In this case, he took diprenorphin, took mu-opioid receptor, purified it, added in heterotrimeric G protein, cooked it with apyrase, and then did an on-rate experiment with diprenorphin in the absence or presence of GTP gamma thiol, and saw a dramatic effect, or an effect. It wasn't dramatic, but an effect on the on-rate. You could also use a nanobody that we had generated against the active form of the mu receptor, and it was also able to slow down the rate of diprenorphin binding. So whether you have a small binding site, like beta-2 or muscarinic receptor, or even an open binding site, G proteins clearly do affect the binding kinetics. And what we view this as is the small class A GPCRs could have a lid, whereas peptide receptors, we think, could possibly just pinch the ligand rather than closing over it. And so some of the residues, parts of the face of the receptor, which may form sort of the fingers that pinch onto the ligand, are highlighted in yellow. And we also can see this very similar conformational changes in the neurotensin receptor. There is a structure of a mutant of neurotensin receptor, which is sort of partially active. And it seems to have, again, these little fingers which may grab onto it. And more recently, a structure from Chris Garcia's lab at Stanford on a viral receptor and binding to a significantly larger peptide agonist, fractalin or CX3CL1, can really show that this is sort of grabbing onto the ligand and binding it with high affinity. And so I'm going to finish that up just by sort of summarizing what we think is going on is that this high-affinity agonist binding site is really due to G-protein coupling stabilizing this closed conformation. And as opposed to the, there we go. Sticky button here. And that the open conformation, the non-G protein conformation, is really accounting for the low affinity site that André Dillon and others had observed. This just, and that this is just in cartoon format, but also that G protein receptors, owing to their basal activity, constitutive activity, can couple to G proteins and lose nucleotide, and also stabilizes closed conformation, and largely accounts for some of our effects on the on rates. I just have one couple of things extra. I can rip through this. So we've been talking about G proteins, but how about other interacting proteins? And one of the most interesting interacting proteins in the GPCR field is its interaction with arrestin, because arrestin is a very nice scaffold and also a signaling molecule. And the question is whether the binding of arrestin is very similar to the binding of G proteins. And a very nice crystal structure came out just recently from Eric Zhu's lab, in which they were able to crystallize rhodopsin with arrestin. And you can see that the conformational changes when you couple either to G protein or to arrestin are very similar. And many years ago, a really elegant study by Seva Gurovich, when he was working with Jeff Benovic, was able to show that arrestin can affect isoproterenol binding just like G proteins do. And in fact, they termed what they had termed at the time an alternate ternary complex. And that we think that this conformational change and the formation of this lid stabilized either by G protein or arrestin would account for that. So this is a long project that's taken many, many years to sort of culminate all the information, and we just recently resubmitted our manuscript. It's currently under review right now. But this work was really done by two main graduate students, Brian DeVry and Jake Mahoney, but of course in tight collaboration with Brian's lab as well as John Steyert's lab. And we thank many people who I don't have time to talk about their contributions, and also the University of Michigan and UCSD for all their support. And I thank you for allowing me to tell us our little story.
I have time for one very quick question. Steve?
Thank you very much for a wonderful talk. I just want to ask about this set of binding curves you've been studying for 25 years. You have one binding site for the ligand, yet you shift the binding curve. It looks, I would interpret that as a change in the relative population
It is.
of high affinity and low affinity sites.
It is.
Okay.
Yeah.
Yeah.
That was quick.
Okay, good.
Thank you.
Our last speaker before lunch is David Comalatti, who's another person who spent time here at UCSD. He's now at the Child Health Institute of New Jersey, where he's an assistant professor of cell biology and pediatrics. And he's going to be talking about some of the neurodevelopmental disorders and molecular bases of those.
Thank you very much for inviting me, Palmer, of course. This is very, as somebody else said before, it's very humbling to be here, to be sort of back home after many years. I spent about 12 years here in San Diego working in Palmer's lab, and I'm now 5 years, been now 5 years since I'm at Rutgers. I will tell you 3 short stories today on what I did here in Palmer's lab, what I did at Rutgers, and sort of the future outlook. And
and then you said they would forward it back with the...
okay, good. So here's the title of my talk. This is the building where I'm currently working on. As I was building this presentation, I realized there was a sort of an underlying thread to my work, and I decided to put it as a subtitle. It's something that many of us know, but probably the younger students need to know before as they work. It doesn't mean that you have to give up what you're doing. You just have to do it. You just have to work hard. So as I was saying, I was a postdoc in Palmer's lab for many years, and I was working in the Neurexin Neuroligand project. This is a picture that nobody showed yet. It was at the EB meeting, and this is one of the many people are here. So there's Pascal and Eve and Scott and Ryan. So was a nice time. So the stories that I'm going to tell you today are something about the Neurexin Neuroligand, what I recently did on latrophilin, and some present and future outlook. So what I mean by synaptic connectivity is something that was 50 years ago was found by Sperry, and what he discovered is that sorry, this is very probably need some water. So what he discovered is that there is between neurons, the way neurons find themselves is through some chemical affinities, and this seems obvious today, but what the prevailing theories back then were something that doesn't even sound in English these days. So what my lab is interested is to study the structure and, when it's possible, the function of cell surface synaptic proteins. And one of the synaptic proteins are populate the synaptic cleft, so between the pre and the post synapse. And they can be of very many different types. For example, they could be transmembrane, they could be GPI, they could be secreted. And they bind to each other in a very complicated way, forming some sort of code, synaptic code, as Tom Sudoff always liked to call it. And there are these adhesion molecules have a lot of very different functions between, for example, neuronal migration and to synaptogenesis, but they also work on, have some non-neuronal function, and in many cases they also have disease-related function, for example, tumorigenesis. And so these are a very important class of molecules. They are on the cell surface, and they can be accessed from the outside very readily. So one of the things that I'm going to tell you about is the structure of the neuroligand. Neuroligands are postsynaptic adhesion proteins, and they have a very high degree of identity across species, but also a very high degree of identity within the family. So much that they have about 30% identity with acetylcholinesterase. And they are expressed in both excitatory and inhibitory synapses, and they have been recently, maybe not so recently now, implicated in autism. So one of the first things that I was working on here was to try to understand the mechanism of binding with the neurexin. And neuroligands are, again, postsynaptic. Neurexin come in two flavors, the short beta neurexin and the longer alpha neurexin that are presynaptically. And their association is basically determined by splice site B in neuroligand, splice site 4 in neurexin, and the association absolutely requires calcium. As we were trying to work on the crystal structure with Pascal and Yves in France, and I was making a lot of protein, purified and send it back to France, but the process turned out to be a little longer than we expected. So Palmer suggested to contact Jill, that I think at that time was working with Susan, to try to have a first look at the structure of the neuroligand through small angle X-ray scattering, which is in some way similar to crystallography. In many ways, it has nothing to do with crystallography. The nice thing about SACS is that it works with a protein in solution, so it doesn't require crystallogenesis that may or may not happen. And after that, data are collected with X-ray, and the scattering decay can be interpreted through mathematical iterations to two-dimensional curves that gives us, for example, the maximum dimension of the particle and an idea of the shape, and also through different type of modeling, for example, ab initio or rigid body modeling, can actually give us information on the three-dimensional shape. These are not really structures. One would probably call them models because they're low-resolution models. Nevertheless, there's a lot of interesting information that can be gathered. So the neuroligands, we could see immediately they were dimers with this kind of shape, and doing the ab initio and rigid body modeling using acetylcholinesterase as a template, we could see the neuroligands were all dimers. When we studied the complex, however, that was a little more challenging because it's a 2-to-2 complex. You can barely see neuroligand here. So we had to resort to neutral scattering to do this, and this is Jill and this is Alex, another collaborator at the time. We had to go to a neutron scattering facility, and using a differential deuteration, we were able to sort of hide the signal of neuroligand and only detect the signal from neurexin. So at this point we had the maximum dimension of the complex, the number of neurexin monomers that we had, and the exact interdistance of the two neurexins. And coupling these experiments with the SACS data, so the X-ray data that instead see the entire complex, plus additional data coming from analytical ultracentrifugation, for example, and many mutants that I had made to understand the interface between neurexin and neuroligand, we were able to create the first model of the neurexin and neuroligand and somewhat place it at the center of the synapse. This turned out to be the cover of the journal Structure at the time, and the first thing that Jill told me once this came out to be the cover is that you better be right. So far, this model is still standing many years after we made it. And so in reference to the title, to the subtitle, neuroligands were discovered in 1995, and no crystal structure was published until 3 crystal structure from 3 different groups came out within 4 days. So this was something that was starting at the time. Fortunately, we were in the mix, and so nobody got hurt, and we were very happy to see that the crystal structure between neuroligand 4, OR1, and urexin done with crystallography and my previous structure done with SACs were fairly similar. We only didn't guess right the rotation of neurexin, but for a low resolution model was sufficiently good. The next logical target was alpha neurexin, because we were previously working with beta neurexin, but this is only a single domain. Alpha neurexin was much more complicated because the architecture was protein was much larger. The architecture were probably more complex. And then again, using SACs, I was able to see the first model of alpha neurexin. So alpha neurexin is composed by 6 LNS domains. And with Megan Miller, there was a previous graduate student in the lab. She took the same protein, but a different approach. And by single particle negative stain, she was able to see that the same sort of clover leaf arrangement on one end and linear arrangement on the other end was conserved using an independent technique. And sure enough, within a few months, but in 1 month, several crystal structures of alpha neurexin were published. And again, we were among the structure published, so we were happy to see that the crystal structure, which are a little different for, we think, a crystallography artifact, but essentially confirm the SACs data that were published before. So when I moved to Rutgers, I was collaborating with Anja van Gogh, and I participated in the discovery of laterophyllin and flirt as ligand receptor pairs. I don't have to introduce laterophyllin because there were enough people talking about GPCR this morning that I don't need to add anything to it. The only thing that what I was working on is on the extracellular domain of this class of GPCR, which is in the adhesion class. So they have a very large extracellular domain. And what we were trying to do was to, first of all, try to isolate the binding cassette, the minimal binding cassette between flirt and laterophyllin. And it turned out there was this olfactomating domain was the only responsible domain for the association with the LRR domain of flirt. They have a very high affinity, below 20 nanomolar, which is kind of unusual for these extracellular proteins. And the association is not ion dependent in any way. It works. It is very tight, and it works in any assay. And also the hydrodynamic characterization just through size exclusion chromatography showed that these are beautiful protein, non-aggregating. So a crystal structure is just waiting to be solved. And when we started doing this, we collected several data sets at 1.6 angstroms resolution. And even with these data sets, my postdoc could not actually phase the data and solve the structure because there was no homologue that was close enough to use for molecular replacement. And so we had to collaborate with Wayne Hendrickson. We got the fortune to collaborate with him. For those of you who don't know Hendrickson, he's one of the fathers of modern crystallography. So it was really helpful to work with him, because he had this recently developed multi-crystal, this sulfur set phasing procedure. Basically, instead of using heavy metal, he was able to phase the data using the cysteines and the methionines present in the structure. So we were able to determine this structure. Remember, This, at that time was a year and a half ago, the first crystal structure of any olfactomating domain. Very interestingly, although the protein is relatively compact, there was one loop that seemed to be very mobile. So we solved the structure of two different forms with this loop that was either up or inside the channel, the pore of the protein. Now the question immediately became, is this an artifact, or a crystallography artifact, or is it true? So in collaboration with Cheng Li here at UCSD, we did hydrogen deuterium exchange. And although this is a little busy and too difficult to see, if we just look at the data for the loop was going to acquire deuterium over a relatively long time scale, so many minutes. It means that it was probably closed most of the time, but occasionally opening. And also interestingly, when the olfactomating domain was in complex with FLIR3, with the partner, there was the deuteration was at this point protected from deuteration. And so when we looked at the data globally, we could not only see that the loop was moving, but all the protected loops kind of came together on the same surface, indicating that this was probably the surface of the interface of the complex. And but there was a previous complex solved just a few months before between ANC5D and another member of the family, FLIR2, that showed that ANC5D bound to the side of FLIR2. But our mutagenesis experiments actually showed that the interface for FLIR was probably the concave face of the domain. So different mutants showed us different affinities, whether or not we mutated something in the belly of this protein. And so when finally the structure was solved, latrophilin, as predicted, was binding on the concave face of FLIR2 with an extensive network of charge and hydrophobic residues in the center of this domain. And again, trying to see if this was an actual biological complex, using a conservation analysis, we found that there is a cluster of conserved residues in the concave face of FLIR2 that exactly mirrors the binding interface, and another conserved phase where ANC5D binds on the side of FLIR. So overall, the mechanism of association for FLIRT and olfactomating domain is that FLIRT is actually a very weak dimer in solution. In fact, in the FPLC that I showed you, there was a shoulder that was resolved by analytical ultracentrifugation, giving us a approximately 40 micromolar affinity. And the olfactomating domain of latrophilin has a loop that allows, I didn't have the time to go into the details, but allows the calcium to exit the structure, and in our case, because of the crystallogenic condition, to be replaced with magnesium. Other olfactomating domains also have a sodium inside the pore, and when they bind, it's a very tight association below 20 nanomolar. The loop closes, and an olfactomating domain binds to the concave face of this solenoid. So then the question immediately became, then is a triple complex possible between ANC5D, FLIRT, and the olfactomating domain? So using ITC, if we use as a titrant always ANC5D, so FLIRT and latrophilin always bind with a very tight affinity. This was published before. FLIR and ANC5D bind with an approximately low micromolar affinity, and ANC5D and olfactomating domain alone don't seem to really interact. But when we use ANC5D as a titrant for the complex, for this complex already formed, then the association, then it does associate with an affinity that is actually a little higher than when it binds FLIR2 alone, indicating that probably there's some of the residues here are actually interacting in this triple complex. So in conclusion, I showed you in this story that the olfactomating domain, it's a five-bladed beta propeller, and there is a unique mobile loop that allows calcium to exchange, although we don't really understand the function of this, and we don't really know the time scale of this exchange, if it is seconds or minutes. And there is a possibility of obtaining a triple complex. We are actually working on this triple complex in collaboration with a scientist in Oxford. Turns out to be a 2 to 2 to 4 stoichiometry, but you guys have to wait to see the actual structure when it's published. Is currently under review. So as I was saying before, we solved the first crystal structure of the olfactomating domain, but by the time we submitted the paper in November, and the time we got published, we are actually not really the first one anymore. So we were number 6 at that point. Structure was just popping up every month. There was a new structure of the olfactomating domain. Again, this protein was first cloned in the 1990s. So it wasn't something that was super hot at this time, but apparently this is how it works in crystallography. And the last 2 papers, so our paper and this from the Meta-Raj, they showed the exact same crystal structure, the exact same proteins, and we did not know that each other was working on this thing. So I don't know how it happens, but. And so now let's go back to synaptic connectivity, and this is something that I've been thinking about and working on for many years now. So as far as I could see between the current literature and several databases, there's about a few hundred protein ligand receptor known at the cell surface for the neurons, with probably 10 new protein ligand receptor pairs discovered every year. And this is probably an optimistic estimate. So I also calculated in using different databases that on the cell surface of neurons, there's approximately 1,500 cell surface receptors. Now, they can be either synaptic or extrasynaptic. There is not enough information to understand, to have this information at this point. So if they have to, if we had to do a screen between these 1,500 genes, it would be a screen of approximately over 2 million binary interactions. And if papers that have been published on the subject show between 1 and 5% success rate for this kind of screen. So we're talking about tens of thousands or hundreds of thousands of protein pairs. And we're now sitting at a few hundred. So how many centuries do we have to go before we have an idea of this synaptic code of the entire connectome? So usually ligand receptor pairs are found by affinity chromatography. Now, I don't know who invented affinity chromatography. The oldest reference that I found, very fittingly, is this one. But this is a very slow process, and usually each lab works on whatever gene they like to is in their interest, so it's a very biased approach. So my idea is that can we define a synaptic code, so go from trying to understand how ligand and receptors bind either at the synapse or just on the surface of the neurons, and go from this relatively simple idea to something that is more realistic, where everything, there is a lot of crowding, there is a lot of protein binding to each other, and some are just bumping into each other, but it is a much more complicated picture. So I was recently awarded a 2-year NSF award to do just this, using an unbiased screen, basically adapting relatively common technology, for example ELISA, to high throughput technology using robotic technologies, and starting with an initial list of 200 genes that will have to be cloned. And interestingly, this type of technique can be applied to many other cell surface proteins, so it's nothing specific to the neurons. And so basically we had 200 genes synthesized in 2 different vectors, so 400 genes. We have an HEK suspension adapter culture running in the lab where you can transfect easily 100 genes in just one incubator and in a 1 ml type of culture, and using a simple ELISA assay, there's nothing high tech here, using a simple ELISA assay, find new ligand receptor pairs and validate them using then independent techniques. And so the system, I'm just giving you a glance, is working beautifully, because with the first 2,000 protein pairs that we found, you can see that in every plate there's something new discovered. And in about 2,000 pairs, we already found approximately 32 positive interactions. 22 of these are unpublished. So, of course, in some of these, we have our positive control. In the neuroligand plate, these are the neurexins, so these are known. But at the same, so we found all the known ones, and we found all the ones, many of them who are currently unpublished, and we validate about half of them. So, because the 200 by 200 screen will allow us to do 40,000 protein pairs in the next few months, we could potentially find a few thousand new ligand receptor pairs. And this may be something that Paul Insel could be interested in for his new screen. So this ties together with what he's doing. But we will have to see. So I have to acknowledge many of the people who did the work that I showed here today. So the people at UCSD here, people around the world that helped me, the funding source, my current collaborators, and of course a picture of my lab as it is now. It's sunny in New Brunswick too. And thank you for your attention.
We have time for a couple of questions before lunch.
It's really, really very nice presentation. I'm curious, because of the extreme similarity of the acetylcholinesterase structure to neuroligand, when you do these kind of last high throughput screens, do you ever find a hit, or did you ever try to use acetylcholinesterase as one to see if there's any hits besides where it's normally found?
Yes. Yes, I tried. No, I didn't find anything. So I was talking with Pascal. She told me, why don't you put acetylcholinesterase into your assay? I did that. In the first 20 plates, which is not much, I did not find anything. The plate is completely clean. But I will keep trying, because this will be extremely interesting to find the extra additional roles for acetylcholinesterase have been described many times, but a new ligand has never been found. So I'm very curious to know that. Yes.
I think a lot of us would be interested in that, because could we find a protein that's got adhesion properties as well as hydrolase properties? See, and we haven't found one yet. It's logical because they're on the back sides of the molecule, as everybody has presented.
So Palmer will be the first one to know if I find a new ligand. But no, Pascal will be the first one to know. But I'll copy Palmer in it. So.
Okay, well, I think we better adjourn for lunch, and we'll be back in 45 minutes. But let's thank all of this morning's speakers.